back to the WashU Virus Genome browser. In this short video, we will be exploring the browser track-based view. To get to this visualization of the browser, users can select the Get Started button in the center of their screen on the browser landing page. Alternatively, if the user would like to have SNV tracks for particular strains preloaded onto the browser, he or she can first go to the data table, add the strains of interest to cart, and then select the Browser View button. When first loading in the browser track-based view for SARS-CoV-2, there are several default tracks that are preloaded. At the top of the screen, users can select the gray box to navigate to a particular region in the genome of interest, either by searching for a gene or typing in a range of genomic coordinates, where the format is the reference strain name, start position, stop position. To the right is a drop-down menu for loading additional data tracks into view. If users select annotation tracks, they are shown an expandable table of tracks that include the genomic ruler, NCBI gene annotations, GC percentages, sequence diversity, mutation frequencies, and comparative genomics tracks. Any tracks that are not already in view can be added by selecting the green Add button. To the right of this is another drop-down menu featuring several applications the user can use on the browser. These include the Region Set view, where the user can view multiple distant genomic regions in one view, various plotting options, session saving for resuming your genomic view at a later time, going live, taking publication quality screenshots, and obtaining the reference sequence for the genomic region in view. To the right of this is a third drop-down menu with settings preferences, which the user can define, which include options for showing the genome-wide navigator at the top of the screen, highlighting a searched for region, changing the highlighting color, restoring the current view upon refreshing, and customizing the track legend width. Beneath these drop-down menus is the genome-wide navigator. Highlighted in green is the region of the genome in your current view. Users can use this panel to zoom into regions of particular interest. Beneath this panel are several tools for navigating the browser, which include the panning mode, allowing users to drag across the genome, the track swapping mode, which lets users reorder the tracks that are in their view, the magnification mode, which allows users to zoom into particular regions of interest, another option for reordering tracks, and several additional buttons for panning across the genome and zooming into and out of different regions, as well as undo and redo buttons. Beneath these options are several annotation and data tracks, the first of which are NCBI gene annotations and a genomic ruler. Next is a track showing the sequence diversity, Shannon entropy, across strains of the given virus species, which only include those from GSAD for SARS-CoV-2. High bars in this track signify genomic regions where the sequence is more diverse across all samples, whereas low bars represent regions with a relatively uniform nucleotide. Beneath this track is a related mutation alert track, which displays the number of strains with the mutation at each genomic position. Next is a track displaying viral RNA expression, and beneath that is a track showing the locations of putative SARS immune epitopes. This particular track is preloaded into view in density mode to allow users to see the abundance of epitopes across the entire genome in one view. However, if the user would like to visualize individual epitopes, they can change the track to full mode by right-clicking, which now displays the epitope sequences. Beneath this track are two related tracks, one showing the locations of transcriptional regulatory sequences, and the other showing the sites of recombination events. Finally, at the bottom is a dynamic track, rotating through showing various viral RNA modifications across the genome. Many of these default data tracks have come from individual pre-publications and are organized into public data hubs. To view all public data hubs currently housed on the browser, the user can select Tracks and then Public Data Hubs. Here, data hubs are organized first by collection and then by hub name. The user can select the plus sign under the Add column to populate a sortable metadata table below. There, the user can either choose to add all tracks from a given data hub into view, or can select specific tracks of interest to add from the facet table. Additional information regarding our collection of public data hubs can be found by selecting the About page linked to the landing page. Thank you.